Hello friends, Jim here with Science Talk. I have a really fascinating paleontological report to share with you. It's a report that was published in the journal GeoBios. The lead researcher is Bertrand Lefebvre, who is at the National Center for Scientific Research and is a researcher at the Laboratory of Geology of Lyon in France. What am I talking about? A 480 million year old creature that's been a mystery has been finally identified from its preserved guts. So the past 150 years, scientists have been engaged in a hot debate as to what this creature is that they have in their archives, in their collections. So obviously 480 million years ago is, a long time ago, well before the Mesozoic. And what's happened was a discovery of a fossil that has stunning details in it, found in Morocco. And this has allowed paleontologists to finally identify what it is they've had all these years. And what the creatures in question are called Stylophorans. And they kind of look flattened and, and armored with a long arm poking off of their sides. So, did it look like a sea urchin, a crinoid, a starfish, brittle star? What it kind of looks something like that. So, scientists didn't know where it fit in because hey, you see this long stalk without much detail, you're going to think a sea lily, a crinoid, something like that. Well, they've now been able to, by examining this new gut find, that they identified it as an echinoderm. Echinoderms include like the sea urchin, starfish, brittle stars, sea cucumbers, feather stars, and those kind of creatures. What's interesting is that echinoderms are one of the few phyla where the organism's body symmetry is radial, not bilateral. I'll talk more about that in a little bit. Okay. So Dr. Lefebvre basically says the findings were made possible thanks to fossils with unequivocal evidence for exceptionally preserved soft parts both in the appendage and in the body of the Stylophorans. These fossils were unearthed in excavation in 2014 at the Fezuata Formation, located along the edge of the Sahara Desert in southern Morocco. And they found about 450 Stylophoran specimens, each dating to about 478, 480 million years ago. At first, they did not realize that the fossils contain preserved soft tissues. That is when you basically go, wow, we hit the jackpot here. As Dr. Lefebvre said, it was only after we unpacked and looked at them under the microscope back in the lab that we can see the soft parts. And the presentations were then later confirmed by a scanning electron microscope. Now this is where it gets really fascinating. The soft tissue finding was unprecedented. Stylophoran fossils have been found worldwide since the 1850s, allowing researchers to determine that these creatures lived from the Middle Cambrian to the Late Carboniferous periods, or about 510 million to 310 million years ago. So this 478, 480 fits right within that time frame when the extinction took place. Because soft tissue is so rarely fossilized, understatement, the Stylophorans are known only from their hard skeletal parts and not from any soft tissues, innards. So the internal anatomy was unknown until these finds were found. So Stylophorans had two main parts, a core body and an appendage attached to it. The core body and the appendage were small, each at about 3 centimeters in length. From there are a bunch of ideas bantered around about what what Stylophorans are all about. From the 1850s to the 1950s, most researchers thought stylophorans were normal, whatever that means, echinoderms. Their weird appendage was interpreted as the equivalent to the stem of sea lilies, as I mentioned earlier. Okay. Now, what's interesting is that this shows why science is really cool. Instead of just sticking to dogma, as new information becomes available, allowing for new improved interpretations, we adjust. Scientists adjust their assessments. It should be. Now, a typical echinoderm has an internal skeleton that's made of mineralized calcitic plates. It is reduced in sea cucumbers, but we won't worry about that for the moment. And they basically have what's known as a water vascular system that helps them to move and as well as respire. Now, you may remember from your high school biology looking at echinoderm and the tube feet, and that tube feet helps them move along, and also the tube feet will then attach themselves around a clam and force open the shell, and then they stick their mouth parts through there and have dinner. So most echinoderms and starfish have a 5-rayed symmetry. They're closely related to another invertebrate group, the acorn worms and vertebrates. Let me repeat that again. Echinoderms have a pentaradial symmetry, penta meaning 5, most of them. Some of the brittle stars have more than 5 arms. They're closely, but they're still radial symmetry. They're closely related to acorn worms, which is a group of, which is an invertebrate group, and to vertebrates as well. And I'll explain why that's the case. It's very fascinating. Echinoderms, acorn worms, and vertebrates make up a group called the Deuterostomia. In the early 1960s, Belgian paleontologist Georges Ubas noticed that the appendage was different from a stem, but similar to a feeding arm as seen in modern starfish. In the late 1960s, British paleontologist Richard Jefferies proposed an entirely different idea. He thought that the Stylophoran main body was a head, holding a pharynx and brain, and that the appendage housed muscles and a notochord. Now, a notochord is a stiffening rod that is a feature of all chordates, and chordates include vertebrates. Chordates, you have the hemichordates, the urochordates, cephalochordates, and then the vertebrates. And if you look at a lamprey doesn't have a skeleton, but it does have a notochord, which is a stiffening rod, basically. 3 features that all chordates possess in the embryological development, and whether they persist or replace, but they're all there, are the presence of a notochord, a pharyngeal gill slit, and a dorsal nerve cord. Invertebrates have a ventral nerve cord. So Jefferies was actually thinking that stylophorans were the link between echinoderms and chordates. Another hypothesis put forth in the 2000s, British paleontologist Andrew Smith basically said that stylophorans were not the quote-unquote missing link between echinoderms and vertebrates, but were more likely primitive deuterostomes themselves, filling the gap between acorn worms and echinoderms. However, this new discovery has helped clarify a bunch of things. The newfound fossils align most closely with Ubas' interpretation, basically the link between echinoderms and chordates. The stylophorans' flat bodies contained intestines, and the appendage was not closed off as a stem would be, and rather looked like a starfish arm. This arm contained a water vascular system that would help the organism move and eat, just like the arms of modern starfish do. Because stylophorans do not have 5-rayed symmetry, they're not radial symmetry, they likely lost it. So, meaning that they perhaps were further along evolutionarily than other pentaradial echinoderms. So Dr. Lefebvre basically states this discovery is of particular importance because it brings to an end a 150-year-old debate about the position meaning where it fits in evolutionarily wise, of these organisms. Dr. Van Rohe basically states that the study is very thorough, and he says, I have no reservations about any of the methods used or conclusions drawn. And as I mentioned, the study was published in the journal GeoBios. So, what is a Deuterostome? Well, to understand what a Deuterostome is, we also need to consider what is a Protostome. So, in order to understand that, we have to go all the way back to the zygote. The zygote, of course, is a fertilized egg. As the zygote develops, it forms a blastocyst, which is basically a ball of the cells. Then the blastocyst further develops into a blastula, where you have a segregation, or the beginning of segregation of the cells, into basically what the cells will ultimately develop into. Muscle tissue, an organ, skin, etc. But when you have the blastula, you have also a blastopore, and then there's the process called invagination, where the blastopore kind of moves in on itself, creating a gastrula. And in the gastrula stage, this is where we actually start to see the three major cell layers: the endoderm, the mesoderm, and the ectoderm. The ectoderm is the outer layer, the mesoderm is in the middle, and the endoderm is the furthest inside. Generally speaking, the ectoderm will develop into the skin and nervous system. The mesoderm develops the muscles, circulatory system, blood, as well as skeletal, if it's a chordate member. And then the endoderm is pretty much your guts, intestines, all the organs, liver, spleen, that kind of stuff there. So now, when you have the formation of the gastrula, eventually you're going to have two openings to the organism: a mouth and an anus. But they do not occur at the same time. One hole, one opening forms first, the second forms later. In the protostomes, which include most invertebrates, okay, insects, mollusks, annelids, you name it. Most of the invertebrate species we are familiar with are protostomes, meaning that the first opening develops into the mouth. Stome, S-T-O-M-E, means mouth. Proto, first mouth. Deuterostomes, it's the second opening that becomes the mouth. So what's the first opening become? The anus. So basically, deuterostomes, which see echinoderms, acorn worms, and chordates, the first opening is your anus. So we basically grow an anus before we grow a mouth. So, not to be crass, we're kind of assholes first. But so that's basically what a deuterostome is. It's the second opening that becomes the mouth. Protostome is the first opening that becomes the mouth. So there you have it. There is a crash course in embryology for you. But this is a very fascinating report and helps explain an ongoing question and mystery that paleontologists have long held. So hope you found this interesting. Thank you for your time. Hey folks, this is Jim here reminding you to please subscribe to my channel and please share my videos with others. Also remember to click the bell so that you know when I drop in a new video. I also ask that you please consider becoming a patron of my channel and support the work that I do by going to patreon.com/Science Talk with Jim Massa, each word separated by an underscore, and becoming a patron. is asking for as little as $3 a month, cost of a cup of coffee, to support the work I do and keep my informative videos coming your way. Thank you for your support.